today, to talk about the treasures of the Linnean shells collections, I'm delighted to welcome Jonathan Ablett. Jon is the Senior Curator in charge of Mollusca at the Natural History Museum in London, where he has been for, I think, for 23 years, we were saying.
Yes.
His current areas of research interest include Southeast Asian and South American land snail diversity and the history of collectors and collecting in malacology. In 2006, he was responsible for the preparation of Archie, which, if you've been behind the scenes at the NHM, is that giant squid. I don't know how many meters it is, actually. It's huge.
8.6.
There you go. Which is the largest spirit preserved specimen in the museum to date. Very impressive. And with that, over to you, Jon. Thank you for coming.
Oh, pleasure. All right, let me just get on to presenting this. One moment. Okay. Firstly, I want to say thank you to Isabel and the whole team at the Linnaean Society for inviting me. It's really an honor to talk to you here and to share some of the wonderful stories and specimens that are in the collection. I've had the pleasure and the honor of working in the collection a few times, and I'm going to talk about that a little bit. And you'll see that it's my name on the talk, but actually, really, I should give some co-credit to my wonderful colleague at the museum, Andrea Salvador. We work very closely together, and she wrote an amazing book called Interesting Shells. And as you heard, most of my work is on terrestrial shells. And unfortunately, I don't think Linnaeus was that interested in terrestrial shells, or at least the specimens in his collection do not represent what I think are the wonderful terrestrial shells. So a lot of the stories you're going to hear, I came from her. And if you want to hear any more of them, her book is certainly worth a read. As Isabel said, I work at the Natural History Museum. Sure, you know it very well if you haven't visited it. We hold around 80 million objects, and of course, the most important being the mollusk collections. We have about 8 million mollusk specimens, so that's about 10% of the whole museum's holdings. And we have around 66,000 type specimens. And don't worry if you don't know what a type specimen is, it's something we're going to talk about very briefly. Somewhat differently from the collections here at the Linnaeum, we have alcohol preserved material, as well as the more traditional dry specimens as well, although probably 7+ million of them are dry specimens. And we have huge engagement with scientists all over the world who come to use our collections. And I don't wish to hammer this home too much, but I often get asked what exactly is a mollusk, even by people who work in the museum on other groups, because mollusks are an unusual and amazingly diverse group. And they're such a diverse group because they've been around for a long time. We've got a fossil record dating back over 550 million years, and this has allowed them to just evolve these amazing forms. I mean, you've got the slugs and the snails, I guess probably the ones we come into contact most with, seashells, but you've also got the cephalopods, you've got bivalves, clams, mussels, and oysters, and then you've got the weird and the wonderful things like the Polyplacophora. You can see at the bottom of the screen this kind of almost woodlouse-looking type mollusk. So instead of having one shell or two shells, they have 5 to 7 plates. Very unusual. And there's even weirder and more wonderful forms. And I guess the kind of the main bits of a mollusk is that they are soft-bodied. They're invertebrates, of course, and they have a shell made of calcium, and they're bilaterally symmetrical. And although we say that they all have a shell, there's always exceptions. And if you think of slugs or squid or octopus, their shells have been internalized or almost in some cases reduced to be almost absent, but they have the kind of evolutionary remnant of a shell inside them. So yeah, inside a slug, inside an octopus, there is a small shell that you can dissect out and study. And because they've been around for a long time, there's over 85,000 species known and probably very many more to be discovered. We get them in all environments worldwide, terrestrial and aquatic. No flying mollusks as yet, but apart from that, in the land and the water, you find them everywhere. So what is a shell? And the Linnaean collections we're going to talk about are shells. And I've certainly, when talking to maybe some school groups, people don't quite realize what the shell actually is. And shells are actually what most people think of if you think of a mollusk. If you pick up a shell at the seaside, maybe you have a small collection of things that you've collected at the beach or in your garden. This is the part of the mollusk that most people are most familiar with. And like I said earlier, not all mollusks have a shell. Some have reduced it, but it's actually produced by the animal itself. It's not like a hermit crab, for example, it's taking a shell actually made by a mollusk and living in that. The shell is produced from a part of the body of the mollusk called the mantle. And they take in calcium carbonate from the environment, be that the aquatic environment or the terrestrial environment, and they mix it up with an organic protein matrix, and they basically add to the shell at the leading edge of the shell. So it kind of grows, and in the case of a gastropod, it grows around in a coiled manner. And this shell provides primarily protection. They are soft-bodied, squishy, and a huge part of the food chain system to lots and lots of animals. So having a shell gives them some kind of defense against predators. And in terrestrial environments as well, it gives them defense against the elements. Terrestrial mollusks always have to be maybe remain moist, so having a hard shell helps them to survive where areas are dry or where there are periods of dryness and help them to keep them moist. And it's likely that this shell evolved in the very early ancestors, probably by secreting calcium carbonate into a kind of mucus that covered it, and that would be taken up by the food, and it basically strengthened the mucus, allowing it to withstand predation from anything in the water around it. And of course, when we think of a shell, I think most people think of a gastropod shell, a snail shell, be that a marine shell or a terrestrial shell. But of course, there are the bivalves, things like clams, mussels, and oysters. And very early on in the development of that shell, it's basically split into two separate shell halves that we call the valves. And I guess one of the striking things about gastropod shells is the twisting, this torsion that gives that kind of classic shell shape. And this probably evolved to save space, to allow more space for organs, but also to reduce the overall amount of shell needed to cover the animal. It's quite a good shape for using less calcium to build a strong structure, which is very important in areas where calcium isn't abundant. Now, what about the shell collections in the Linnaean society? Well, when I was kind of looking at this talk and trying to build up a history of it, there were three main resources I used. And the most well-rounded, the most in-depth kind of Bible, I guess, for looking at this collection is the report from the 1960s by Peter Dance, who previously worked in the Natural History Museum. And if you want to learn more, there is nothing better that I would recommend. It's very in-depth, but absolutely yeah, a fascinating read. Then Kathy Way, who was previously actually one of the curators responsible for the collection here. She wrote a shorter article, maybe kind of a précis of Peter's work. And then, of course, there is the Linnaean's website again online here. So if you're interested and want to learn more, I would recommend any of those resources. So the shell collections here at the Linnaean are pretty large. It's around 588 lots, representing 550 of the more 800 species he named. So there was about 700 species named in the 10th edition of Systema Natura. Then in the 12th edition, there was about another 100 that he added. And then in later publications, I think there was another 28. So yeah, about 828 or so species of mollusks that he described. And you can see just over half of them are represented in the collections here. And of course, we have to, when we look at the collections here, we have to also think about the collections in Uppsala, where there are another 1,000 odd lots with Linnaean provenance. And when we look at the collection as a whole, most of the shells originated from places that Linnaeus had never visited. So therefore, we have to presume that he obtained them from other people, maybe through purchasing, maybe through swapping them with colleagues. We probably will never know, but we know that he didn't collect them himself. And there was a really lovely thing that came out in a biography of Linnaeus by Jackson in 1923, where he found a previously unreleased letter where it said that Linnaeus's collection was valued at $12,000, which apparently is where the etymology of the word dollars comes from. It was a term for a Scandinavian currency at the time. So that'd be about £250,000 today. So a substantial collection. And after Linnaeus's death, his son Carl actually, we know that he added specimens to Linnaeus's own collection, and he obtained them from people like Joseph Banks, who, of course, was on the Endeavour and a famous botanist in his own right. Daniel Solander, who was a pupil of Linnaeus, it was also with Joseph Banks on the Endeavour, and people like the Duchess of Portland, who were very well known and respected shell collectors. She is thought to have one of the largest and most impressive shell collections of the time. And then when James Edward Smith purchased Linnaeus's effects, including the shell collections that we're going to talk about, we know from the documentation that Smith is known to have given away specimens. There are reports from exhibitions in books that other people obtained specimens from James Smith that that had provenance to the Linnaean collection. But also we've learned that he added to it as well. He wanted to make sure that Linnaeus's collection remained up to date. So he wanted to add newly described species, rare species that he was able to obtain into the collection. And it wasn't really until the 1840s when Sylvanus Hanley really tried to make sense of the Linnaean collection. And some have said that in some respects he may have made it worse. He went on to publish his 1855 work on it, and I actually think it was quite a valiant attempt, a great starting point to kind of disentangle some of the problems with the collection, to try and work out exactly what specimens Linnaeus has had, and to kind of suggest how he used them. But he did things that today would be a real curatorial no-no. He kind of separated specimens from their original labels. He glued some of them onto wooden tablets with blue paper. So there were definitely ways in which he made it more difficult to try and understand the collection as Linnaeus had it. A bit later on, Henry Dodge, a US malacologist or mollusk researcher, published on the collection, although I don't believe he ever actually saw the collection himself. He did it from remotely and through letters. But as I mentioned earlier, it was in the 60s when Peter Dance made a really thorough review of the collection. And you can see in the picture on the screen that he made these beautiful cards that went with every single lot, where he would write where they were listed in the Systema Natura, but also reference Hanley and Dodge's works, and then give his own thoughts on each one. And a project I'm going to talk about shortly, these were unbelievably useful. Peter Dance really immersed himself in this. He really, kind of really made the best effort of trying to untangle a very difficult collection. And as well as these little notes, there were much longer, long form, typed up sheets that went with the collection as well, a really valuable resource. And he noticed things like some of the larger, more beautiful, maybe collectible specimens seem to have disappeared after Hanley had finished compiling his manuscript. What happened to them, we may never know. But it's interesting to see that things like the strombus, which is illustrated below, is a really beautiful seashell, can't be located in the collections today. And Dance described the kind of various states, is a polite way of putting it, of curation of the Linnaean collections. There was loose specimens in drawers. There was Linnaean metal boxes, these metal tin coated steel boxes that were found in the collection. He found blue card trays that he thought had been brought in by James Edward Smith. He found more modern glass top boxes, the shells glued onto wooden tablets, as we've spoken, and also, and what I love, is the assorted paper packets, pill boxes, and chip boxes. And working at the museum, I see this all the time, and whilst not good curatorial practice, it's always exciting when you find something that is still in the container it was collected in, a matchbox, an old pill box, something of the time, the day, maybe with a bit of newspaper in or something, padding it out, something that gives it a kind of sense of the time and place it was collected with. And Dance really kind of hammered home just how confusing this collection was and how much care people had to take when analyzing the collections. And he thought, because we couldn't be sure of the provenance of a lot, you had to be really careful when evaluating type material. And I suppose this is a good idea just to say, what is a type? So if you remember in my introduction, I said that at the museum in London, we have around 66,000 type specimens just in molluscs alone, which is a fairly large number, one of the largest type collections for molluscs in the world. And this type material is what most scientific visitors to the museum want to see. And it's usually considered the most scientifically and historically valuable material in any natural history collection. And so type specimens are the specimens that are, the easiest way I think of it is the specimens in front of an author as they described a new species. So when you describe a new species, there are lots of things you have to do to make it valid in the eyes of science. You have to name it, you have to describe it, and then it's very strongly recommended that you have an object that fits that description and that name. And these are what we call type specimens. And when I tell this to students, especially, they don't seem that interested in what is a type. And I get a feeling that it's because when we think of animals, we often think of common animals, animals that are very familiar to us. You know, you might think of an elephant, and it's very easy to understand in our brains what an elephant is. When you learn that there are two species of elephant, it's quite simple to work out the differences between them. So we know there are two species of elephant currently alive, and they have two different names. But when you talk about animals that may be less familiar, maybe you're talking about micromolluscs, maybe you're talking about insects or something, the differences between these species can be very slight. They may be only really discernible by an expert in that group. So having the specimens attached to the name and the description help us to understand often many, hundreds of years later, exactly what it is that this author meant when he described this new species. And these specimens, these types, are linked for all time to the name and to the author and the date when these species were described. And of course, Linnaeus created this system of nomenclature, of naming, that we still use today, an invaluable system. But he didn't set up the type concept. And it wasn't until 1961 when the International Committee for Zoological Nomenclature standardized and regulated this system that we had a way that we could agree how we should name, how we should describe, and how we should deposit type material in order to make sure that we were all doing things the same way. And this is a nice little side segue to some of the work I've been involved with the Linnean Society molluscs. I've been lucky enough to visit a few times, but probably the longest I spent here was with Mollusca Types in Britain and Ireland project that was funded by the Jon Ablett Foundation. And this joined the mollusk staff at the Natural History Museum in London with those in the museum in Cardiff, Wales. And basically, we had the idea to go to all the larger regional and national museums across the UK that house significant mollusk specimens, and more importantly, housed significant mollusk types. And in many of these institutions, there might be one person, two people who look after all the natural history objects, or sometimes all the natural history objects and all the ethnographic objects, the musical instruments, everything, and often aren't specialists. And the aim of this project was to search and to make available the types in each institution, and to give some training, support, and a website for the staff at these institutions to display their types, and also as a portal to help with inquiries and the like. And of course, we couldn't not include the Linnean Society. So back in, I think, 2019, we spent many long and lovely hours going through the collection, analysing each of the specimens, building on the work that Peter Dance had done, adding more up-to-date data. And this is all available online through the website that you can see at the bottom of here. So if you're interested, please do go and have a look. So that's kind of my introduction to what is a mollusk and a bit of a history of the collections here at the Linnean. But obviously, the best bit, the meat of it, is talking about some of the amazing specimens that we have here. And it was actually I found it really tricky to decide what to talk about. I only have a limited time. There are 550 specimens, and how was I going to talk about them? And in the end, I kind of thought I wanted to talk in three different ways about the mollusks. The first is how humans have used shells over many years. Then I'm going to talk a little bit about adaptation to environment. And then lastly, just highlight a few of the struggles that mollusks are facing due to the ongoing biodiversity crisis. So to start with, shells, humans have been kind of, I guess obsessed might not be the right word. Maybe I'm obsessed with shells. Humans have been fascinated by shells since all of recorded history. I think one of my favorite facts is that the first, well, debatable, but what is often thought of as the first piece of artwork was made by not even our own species, but it was made by Homo erectus on a bivalve shell about 500,000 years ago. That's about 200 to 300,000 years before our own species came into being. And this kind of engraved bivalve shell could be thought to be the first piece of human art. Doesn't matter if it's not quite, how we think about that, but I think it's quite an interesting fact. And if you think about human history, you might walk around a city and see shells engraved on buildings and on churches. Many religions have incorporated shells into religious practices, into architecture, into symbolism. If we think of kind of art, shells often appear in them. The golden ratio of the spiral is often seen in architecture, in art, in engineering, and people have used shells as objects, as musical instruments, as forms of money, as jewelry, as decoration. And I don't know why this is, but I'm sure it's something to do with the spiral form of gastropod shells and just how, I think, pleasing they are. This golden ratio, this outline that somehow kind of captured the imagination of humans for such a long time. And this is one of my favorites. This is Triton's trumpet, Cheronia tritonis, as it's now called, as described by Linnaeus. As of course, all of these ones I'm going to talk about are described by Linnaeus. And we know that this has been used through many cultures in early human history as a trumpet, as a musical instrument, or as a way of making a loud noise. We know that it was used as a war horn in sort of North Africa, being able to kind of, I guess, startle your enemies just before going into battle. When specimens of this species, which were traded or moved into Europe, they were used by farmers as a way of calling to. We're not sure if it was there to animals, to alert animals, or more like to alert other farmers in rural areas of their presence or of danger or possibility. And they're used in different cultural symbolism as well. So in Hawaii, they're often played at sundown in ceremonies to welcome the night and to celebrate the end of the day. And in Japan, they're often used in fire cleansing rituals, the climax of one of these displays. So you can see different societies all been sort of enamored with this one species. I'm just going to stop sharing and then hopefully move the shell here. This is one of the most beautiful, I think, shells in the Linnean collection. And you can see, I think, just it's the interesting shapes, the ridges, the color. It just really is a beautiful object. And I was saying before how shells grow. This is the leading edge of the aperture. So as the shell grows, they're taking calcium from the environment and you're laying it down here. So as the shell grows, it'll get longer and continue to spiral round from this edge here. So yeah, a really beautiful shell and a lovely story behind it too. Now I'm going to show you a glimpse of something far less beautiful, but with a possibly more fascinating story before. So I really wanted to talk about this despite this sadly being the extent of the specimen. So this is a small fragment of Pinna rudis from the Pinnidae bivalve shells. And I realized in my presentation just now that I haven't actually got a picture of the whole specimen, but these are a large sort of 20 centimeter long bivalve, sort of fan shaped. If you imagine a kind of fan, so very narrow at one end and splayed at the other, and you might just be able to make out the ridges on this shell. So it has a kind of peaks and troughs along the whole of the shell form with kind of these shell prongs on the outside. But it's not the shell itself that I really wanted to talk about. It's actually what humans have done for them. So since about at least till 4 AD, but possibly much earlier, it's possibly thought that the Egyptians produced cloth in the same way. This is a substance called sea silk. And sea silk is a beautiful gold color that's produced by treating the fabric with lemon juice. The sun is used, but this material actually comes initially from the byssus thread of Pinna rudis and the closely related Pinna nobilis. So Pinna rudis lives in the Mediterranean, but also in the Caribbean, the Black Sea, Gulf of Mexico. Pinna nobilis is just a Mediterranean species. And if you've ever bought mussels or other bivalves, maybe from a fishmonger, or if you've had them in a restaurant, sometimes they have this kind of hair-like structure on the thinner end. And this is called the byssus, and it's used by the animal to attach itself to the substrate, stop it moving around. And the Phoenicians, or possibly even earlier, worked out that by processing this byssus thread, they could produce this wonderful golden thread. And it's often been suggested that the legend of the golden fleece could have come from this golden material. It was incredibly costly to produce. It was thought that around 1,000 adult specimens were needed to produce about a kilogram of fabric, which made far less yarn when it was actually spun. So it was only reserved for the very wealthy members of society. The gloves in the picture actually belong to Hans Sloane, whose collections formed the foundation of the British Museum. Queen Victoria had a pair of stockings made out of them, and the rest. And it's known that there were certain popes that had special ceremonial gloves, stockings, slippers made out of them for certain religious ceremonies. I believe there is one family that still produce thread in this way in Sicily, but it pretty much died out with the introduction of much cheaper silk as the silk routes opened up and silk was more readily available across Europe. But yeah, I just love this story, and the pictures don't do it justice. This really such a vibrant, beautiful cloth. So the next specimen I'm going to talk about as well is also related to cloth and clothing, but this is actually to do with the dye. So this is a muricid, Balinus brandaris, shallow water. You get it around Mediterranean, North Africa. They get to about 20 centimeters in size, and they have an unusual method of self-defense. And that is that when they are disturbed, they produce a chemical into the water that is possibly distasteful to predators, but it also turns bright purpley red in the presence of sunlight. And humans around just before 0 BCE realized that they could use this in order to dye their fabrics. And purple being very rare in nature, it was almost, instantly awarded this kind of regal royal state. And that's probably one of the reasons we often think about royal regalia. It's often this kind of deep purple because it's such a difficult material to make. And again, it needed many thousands of individuals just to get a very, very small amount of dye. And again, if we go to the camera here, you can see two examples of the shells. The spines you can see here used to anchor the animal into the sediment to stop it moving around with the tides and the underwater currents. And really nicely, you can see here in the aperture of this one of Linnaeus's numbers that he used to order his collection. One of the things that Peter Dance would have used in order to verify just where some of these specimens could have come from. And if we pop that back, the next thing I wanted to talk about is a little bit smaller and maybe a little bit less glamorous. But this is, and I'm going to hold this one up because it's a little bit smaller. This is Crepidula fornicata. And these exist in the water. Originally, we were from kind of the coast of North America on the East Coast, sort of between Mexico and Canada, so sort of central US. Although they have been moved since the 1950s into the waters around the UK and around Northern Europe as well. They're a bit of an invasive problem. And I wanted to talk about these not really because of their use with humans, but more about their reproductive life cycle. But actually, before I go to the back to the PowerPoint. This is a lovely example of one of Linnaeus's tin boxes that he had made for the collection, all the same size, 5 centimeters by 5 centimeters, the little space on the front for labeling. Now, interestingly, when I see this in the collections in London, we have the collection of Joseph Banks, and he had very similar boxes made. And I often wonder if they, did they collaborate on them? Did they get inspiration from one another? Did they get made by the same people? Who knows? But yeah, the way that you get these same kind of forms through different collections, I think it's very interesting. But if we go back to the PowerPoint, you can see some of the specimens when they're live. And the reproduction of mollusks is really fascinating. There's lots of different strategies. In some groups, like the cephalopods, you have separate sexes. So you have male individuals and female individuals. In other groups, like terrestrial land snails, you have hermaphrodites. So they have male and female sex organs. Some can mate with themselves, some cannot. But Crepidula fornicata are what we call sequential hermaphrodites. And that means that you start as one sex and over time you change into another form. And with these, they start off as male. And the reason for this probably is because biologically sperm is cheap to make. It doesn't take as much resources as the eggs. Also, as they get larger, they're able to brood more eggs. So they start as male, and over time, as they grow, they gradually turn into female. And this larger body size allows them to brood far more eggs. And it's thought they can brood between 400 and nearly 1,500 at any one time. And then so you've got these small males at the top, and then larger and larger specimens until you get to the larger females at the bottom, which I think is, yeah, a really interesting way, an unusual way possibly of passing on your genes. And a lovely fact that I learned the other day, actually, if you just go to the next slide, is I was thinking about the name Crepidula fornicata, and I wondered if it referred to the fornication, the mating behaviors, the unusual, but apparently it comes from the Latin word arch. So apparently fornix is Latin for arch. And in Roman times, the prostitutes would gather under arches to sell their wares. They became known as and related to brothels. So apparently that's where we get our word for fornication now. It comes from arch. So there's a lovely little side fact for you as well. Now, the next one I want to speak about was Conus geographus, and I've put him in the wrong place. Where have I put him? I've moved it. Oh, here. So cone shells are really fascinating. They've come up with an amazing way to catch their prey because different mollusks eat different things. So some are herbivores, some are detritivores, they eat kind of rotting material, and others are active predators. And the cone shells are active predators. There's two sort of subgroups within the cone shells. There's the ones that eat worms and the ones that eat fish. And I guess one of the famous things about snails, be they in the water or on land, is they're slow. And where these live in the Indo-Pacific on coral reefs, there's a lot of competition for food. If you don't catch and eat your food, something else will grab it and steal it off you. So how does a famously slow species get its prey? Well, all gastropods have a structure called a radula. It's also found in the cephalopods as well as an aside. And you might have seen this when you've seen a garden snail kind of rasping off your lettuces. They're using the radula to scrape the top of the vegetation. In the cone snails, they've evolved to have their radula like a harpoon or a syringe, I guess. And this is fired into the prey. And when it's done so, it actually injects a huge cocktail of toxins into the prey item, be it the worms or the fish. And this instantly paralyzes the prey, meaning that it will hopefully fall exactly next to the snail and it can kind of engulf it and swallow it before anything else can get its hands on it. And each species tends to have a slightly different form. You can see on the right some of the different forms of these harpoon-like radulas. And I guess there's a human interest angle to these as well, because this cocktail of toxins is very interesting to the pharmacy industry. So I know currently there are 2 drugs that have been produced based from the toxins found in this venom. One is sort of an end-of-life pain care medication, and the other is for treatment in severe epilepsy. because obviously one of the things that one of the toxins does is stop muscles from activating, and this can be used to calm tremors in extreme cases. And we have one here, again, a really beautiful shell. Love to show you. Again, you can see Linnaeus's number in. One of the strange things I always think about this group is that, you know, the shell is almost completely covered by the body in life, as you may have seen from the pictures, yet they still have this amazingly ornate shell. And this is called the geography cone. And I've always assumed it's because the patterning you can see around the shell makes it look like a kind of a map or an atlas. It's actually much clearer in other specimens I've seen. But you might get an idea of the kind of, the patterning that resembles a map, hence the wonderful name. So the animal is inside here, and this is the business end where the eyes and the harpoon and the mouth come out of here. And my next specimen to show you is actually a group that I do work on. As I said, I don't really work on marine gastropods, but I do work on cephalopods. And there are two similar but very different species I wanted to show you today. So these look very similar in form. Actually, I'll hold them a bit higher. And you can understand when they were originally described and studied, people thought they were very closely related. In my left hand, on the left of your screen, is a paper nautilus, and in the right is a chambered nautilus. But actually, with our current understanding, we know they're pretty much as distantly related within the cephalopods as they can be. And it turns out that this is not a shell. So the nautilus, the nautiloids, of which are about 7 or so, we're not quite sure, species, are the only group of cephalopods to have retained an external shell. So the squid, the cuttlefish, and the octopus all have internal shells, whereas this has an external shell. So they have around 90 tentacles, and the animal sits in this last chamber here. Its tentacles moving out here, and it moves up and down the water column. They occur in the Indo-Pacific, down to about 200 meters at night, coming to the surface to feed. And they move up and down by exchanging water and gas that are in these kind of sections that you can't see, sealed off sections. It moves up and down, I guess, like a submarine. So that's your chambered nautilus. Whereas it turns out this is actually the shell. Well, it's not even a shell. It's the egg case of a type of octopus. And actually, if we go back to the PowerPoint, you can see a little bit better how this works. So on the right, you can see the lovely nautilus, the chambered nautilus, and you can see the large eye. A really old group. They've been around for about 400 million years ago. And actually, when you see fossils from about 300 million years ago, they're very similar to the ones we have today. And on the left, you can see this basically octopus, which lives in the case. And it's only the females that produce this egg case. And they use it to protect their eggs, to stop them being eaten by predators, to keep them close to them. And they kind of produce this case by a gland on the fins. And she's just pressed herself in there to keep it. She's not actually attached to it. And the males look very, very different. They're what we call having extreme sexual dimorphism. So the bottom is the male, looking very different than the female on the top, much, much smaller. And in fact, for a long time, no one could find the male in life, but there was a species of parasitic worm that was described being found in the egg case of the females. It turns out this is the discarded pseudopenis of the males. Cephalopods have weird and wonderful mating. They have a structure called the hectocotylus, which is basically a way of transferring sperm from the male to the female. And his, for some reason, grows in a pouch below the eye. And when it's time to mate, it kind of is released from the pouch under the eye and almost swims into the female, where it delivers the sperm. And yeah, when this was first discovered, scientists thought it was a parasite and not the male member of a Argonaut, which I always think, yeah, it's kind of fascinating the way we look back on things. And then if we could go to the next slide, this is an absolutely bonkers shell. I don't really know much about this. There isn't much in the literature, but these are possibly the strangest. This is a bivalve, believe it or not. They're also called a watering pot shell because the larger end looks a bit like a spout on a watering can. And that end is buried into the sediment. and the narrow end sticks out where they can filter feed. But what happens is when they hatch, they're actually hatch and they look like a normal bivalve. You can see in the middle picture the kind of remnants of this bivalve shell within the larger structure. But as they grow and mature, they secrete this calcium tube around to protect it as it burrows into the soil. There's very little known about this species. It's just not very well known, but just an absolutely, I think, fascinating form. And if I show you under the microscope, I mean, this is when I've had students in, I often try and please divide up these shells into the different groups. You know, could you divide these into gastropods, into bivalves, into cephalopods? And this is the one that they always get wrong, and understandably so, because it really doesn't look much at all like a bivalve. And you can see on the end this kind of watering can end as well that gives it its name. And lastly, I just wanted to highlight a few of the specimens that have been really kind of at the mercy of the kind of mass extinctions and biodiversity crisis that is going on at the time. And I'm going to go back to land snails here because that's my default, because there have been more recorded land snail extinctions in the last 200 years than there has of any other group of animals that we know. That's not to say that other groups haven't gone more extinct. It's just the recorded ones we know, there's been more land snails. So molluscs are not immune. It's not just the beautiful big cats, elephants, and whiskery things that need our help. It's also the molluscs too. So this is a very small giant clam. So giant clams can grow up to about 130 centimeters in diameter. They get to over 200, maybe up to 250 kilograms, found across sort of Indo-Pacific tropical waters. And they have a really interesting life strategy. They don't feed directly from the environment, but what they do is they harbor algae, zooxanthellae, and these photosynthesize, so they open up in the daytime, allowing that, and then they kind of grow, and then the animal itself actually digests the algae and feeds off those. So it feeds off what it is protecting. So there's a kind of trade-off. It can't take too many or it won't survive, but it needs enough to be able to sort of regenerate and produce the next generation of algae. These are taken commercially in some parts of the world for food. So there's a lot of exploitation of this. Also taken for the tourist trade, although they are CITES protected, so it's an illegal trade. But also with global warming, changing the environment. Threat from extinction from climate change. And the last specimen I wanted to show you was actually the oyster shell, which is on the far right. I'll just get that 1 second. We could actually, yes, you could show the slide at the same time. Last one. So you can see this again. These animals look much like nicer in life, and you can see the mantle tissue where they house the algae that allow them to get nutrition from the environment. But for my final specimen, if we go to the last slide, it's not just in far-flung tropical places that there is problems for molluscs. There's lots of species endangered on our own doorstep. One of them being the native oyster. And oysters have been harvested by humans for a very long time. There's records going back, to the very earliest times of humans. And in fact, with the Roman occupation of Britain, the Romans loved it. Our native oysters so much that they would ship them back in the thousands to be enjoyed in Rome. Unfortunately, this species is in quite severe decline within the UK. It's very sensitive to changes in temperature, in salinity, to habitat degradation from things by trawling. It's very at risk from non-natives who may change the environment, making it less suitable for them. And so their populations have dropped drastically, and it's actually led to people bringing in the Pacific oyster from America, which is harvested and is actually a lot more productive, so better for the trade. But again, it's not good for our own native wildlife and for this species. And again, just to finish off, I have this beautiful oyster shell. And you can see, just this kind of typical oyster shell motif. Every oyster is different, which I find amazing. I actually don't know how you get such these different forms and what it is about the environment that causes these changes. If it's
the differences of water currents, salinity, temperature, all the substrate I imagine that they're housed on as well. And that's been my kind of pick of my favorite mollusks in here. And yeah, I'm very happy to take any questions that you may have.